speaker is Mohsen Sadeghi, and he did his PhD with Daniel Walport at Cambridge, and he's now a postdoc in the Action Lab with Dagmar Sternard. And today he's going to talk about merging control strategies for the manipulation of complex objects. So over to you, Mohsen.
Thank you very much. Can you all see my screen? And is my voice clear? Okay, I assume so. Thank you. I'm very pleased to be here, and the same session as my lab members, basically. And I think I'm going to have a very easier job explaining after Martin and Reza. So I'm Mohsen Sadeghi, as Nidhi mentioned. I'm a postdoc at Northeastern University, and I'm mainly interested in the control mechanisms underlying that manipulation of complex objects. And in particular, I'm very interested in rhythmic and discrete movements and their interactions. So when we perform movements or manipulate objects, depending on the goal of the task, we might use different types of movements. So in swirling a glass of wine, the main goal of the task is to bring the wine into a circular rotation. Whereas when we bring the glass to the mouth, the goal of the task is to translate the glass itself without spilling the coffee. And there's been a lot of questions and a lot of studies as to whether these two types of movements arise from fundamentally different control mechanisms. And fMRI studies in human have shown that different but overlapping regions of the brain are activated when performing rhythmic versus discrete movement. And when learning, people have shown that the transfer of learning from rhythmic to discrete and vice versa is not a symmetric transfer. And lately, people have found that even distinct functional modules, the motor cortex of mice, is responsible for rhythmic and discrete movement. So it seems that there are separate mechanisms underlying these two types of movements, but these mechanisms perhaps are interacting. And that's because in our real life activities, we often perform complex movements, which are not purely rhythmic or discrete, but a combination of both or a sequence of one after the other. And the question is, how do we smoothly transition between rhythmic and discrete movements when we perform complex movements? And we're mainly interested in that question in the context of manipulating complex objects and in the real experimental setting as opposed to virtual. So Reza talked about the virtual setting where he studied the cup and ball manipulation. The importance of studying real interactions with objects is that there are real sensorimotor consequences, and these consequences could actually affect our decisions. So if you make a sudden movement when carrying a cup of coffee, you may have to deal with real trouble. So I think it is very important to basically move towards studying more realistic experiments. Now in the lab, we don't really work with a glass of wine, merely because subjects just get bored and start to drink it. So instead we replace it with a cup, simple cup on horizontal plane with a ball rolling inside. And here is the experimental setup, a table with a smooth surface and the custom designed cup on top. Subjects can basically slide the cup onto the surface easily. And on the top of the table, we have a camera which records the movement of the object, and we use shape detection and color detection algorithms to basically measure the position of the object and to detect the shapes on the table which define the task. So the task that we design is fairly simple. So subjects start with rotating the ball in the cup while in a home position, similar to swirling a glass of wine. And then auditory cues introduced, after which subjects are instructed to translate the cup from the home position to the target position. And in this translation, they have to try to keep the ball rotation as steadily as possible and try to avoid the ball from falling out the cup, which is a possibility. And after they arrive at the target, they have to restore the ball rotation as steadily as possible again. So here it's me sort of demonstrating the task. We start with the rotation of the ball, the home position, and then the translation to the target position. And basically we go back and forth between the two. So we collected data from 5 subjects. This was basically right before the pandemic. And each subject performed 8 sessions on 8 different days. And we had 150 trials per session. So in the end, we ended up with 1,200 trials for each subject. And I'm going to basically show the data from all subjects in this study. Here we have an overview of the trajectory of the cup and the ball with respect to the inertial reference frame. I'm showing here about 100 example trials, and the black line here is just a single example trial. And we're going to mainly focus on the movement of the ball. So we can take the ball trajectory and represent it in the cup frame of reference and basically break it into 4 different sessions or 4 different phases of the trial. So at the rotation phase, you have a very fairly stable rotation of the cup of the ball, and then there's a go cue signal that's introduced, and then the translation is initiated here and then ended here, and then the recovery basically when they restore the ball rotation. And we can look at the ball position at the critical stages during the trial, which are mainly at the go signal, when they start the transition, the translation of the cup, when they end the translation of the cup, and basically the movement time from the start to the end of the translation. We can also introduce a performance index to see how well the subjects were performing. And that's essentially, we can take the ball trajectory with respect to the cup, and at each point we can take the distance of the ball from the center and plot that distance as a radius of the ball position against the time. And as you can see, that the distance is fairly stable at the rotation phase, and we can basically take the average of that and take that as the reference radius for the rest of the trial. So any deviation from this reference circular movement is essentially the error of performance. So we can basically represent as a root mean squared error of performance. So if we look at this sort of error for each subject here on the vertical axis, I'm plotting this error across the trials. Each dot is one single trial, and the solid line is a running average with a 20 trial window. And 3 of the subject, 1, 3, and 4, basically show very stable performance in terms of changes in their performance index. So there's no change in the performance index. We see learning in subject 2 in a peculiar way, where he basically figures out how to do it, and then the performance improved instantly, and we have a gradual improvement in the performance subject 5. And overall, if you look at the moving standard deviations, as a measure of variability, variability goes down for most part, which is an indication of learning. But we're mainly interested in the transitions between the rhythmic and discrete movements. So how do we sort of look at those? Well, essentially, what we can do is we can look at the position of the ball with respect to the cup and represent it as angle or the phase of the ball with respect to this horizontal line. And we basically measure that angle for different critical stages of the trial. We can basically plot the histogram of those angles in a polar histogram such as this. So we can start basically with the phase of the ball at the go signal, and we plot the histogram of that. We basically find a very fairly uniform distribution of all the angles across all the trials. And this is not really surprising, because at the go signal, subjects are not really expecting the go signal, so they don't have any control over the phase of the ball. And if you fit a simple fun resist distribution to this data, we can basically estimate the concentration value here. It's just a little sidetrack. So if you have a fun resist distribution, the concentration value is essentially analogous to the inverse of variance. So concentration of 0 means a uniform distribution, and as you increase it, you get more concentrated distribution around a certain angle. So the concentration value is very small and close to 0, meaning there's a uniform distribution when the ball for the ball at the go signal. But what about when they start the transition from rhythmic to the discrete movement? If we look at the distribution of that, we can see that there's a clear modulation of the ball phase when they start to transition to the discrete movement of the cup. So this sort of discrepancy between the go signal and the start of the discrete movement shows that subjects are actually actively modulating the phase of the ball in order to basically have smooth transition towards the discrete movement. So how about when they basically end and arrive at the target? So we can look at the distribution of the ball phase at the target, and we can also see a very concentrated distribution around a certain angle. Now, this might be different across subjects, but also within subjects, they're all very consistent in the angle of arrival. Now, we know that they modulate the ball phase when they transition from rhythmic to discrete movement. But can we look at this plot, which is the target, and say they also actively modulate the ball phase when they arrive at the target? Because another null hypothesis would be that subjects just choose one initial angle at the initiation of the translation, and they basically play out the discrete movement, and it just happens that a ball arrives at a consistent angle every time. And so this is not really what is intended by subject. It's just a product of choosing the right initial angle. And to answer that question, basically examine that question, we can just look at the translation time, where subjects basically translate the cup from home position to target position. And if we plot the translation time against the ball phase at the initiation of translation, we get a polar plot such as this, where the angular dimension is essentially the ball phase at the start of the discrete movement, and the distance from the center is the movement time, the translation time. And we plot those against each other, you see this interesting spiral pattern which indicates a linear relationship between the time and the initiation phase. And that is basically the case for at least 3 subjects, but not so much for 2 other subjects. So what this means essentially is that if subject starts with an initiation angle, let's say here, the movement time has a bimodal distribution. So they either arrive at the target at this time, or they delay the arrival for this amount to arrive at a later time. And this difference between the two basically happens to be close to the close to the period of one cycle of the ball rotation. So what this indicates is that subjects are sort of integrating the discrete movement into the rhythmic movement of the ball rotation when they basically transition back from the discrete to the rhythmic movement. So this indicates that not only they modulate the ball phase when they transition from rhythmic rotation to the discrete, but also when they transition from discrete back to the rhythmic. So the question now is, what is the advantage of a spiral sort of pattern like this? So to answer that question, we did a very simple simulation where we simulated a cup and ball rotation with a sinusoidal movement of the cup. And then at a given ball phase and a given time of the given movement time, we translated the cup with a minimum jet trajectory to the target position. And then we basically measured the position of the ball and its deviation from perfectly circular motion. So here I'm basically showing an example simulation where you start with the rotation of the ball, then the minimum jet translation, and the end we basically measure the deviation of the ball from a perfectly circular movement. And with that, we have basically independent variables where we change the translation time within this range, and then the ball phase within this range. And at a given phase and time, we calculate the error of performance. And we basically plot the error of performance as a function of these 2 variables. We get a solution manifold, which looks like this, where, sorry.
Sorry, if you can just wrap up quickly, we have a couple of questions for you because this session ends in 3 minutes.
Oh, okay. Yeah, it's the last slide, or second to last. So we have basically the initiation phase, the movement time, and the color is basically the color gradient is the error, the normalized error of performance. And the red dots basically indicate the cases where the ball escapes from the cup, so the ball falls out of the cup, so they're not really accessible. And if you look at these low error areas, these dark areas, you can see that there's a similar spiral pattern emerging from these simulations, which resembles the performance of the subjects of some of the subjects in the experiment. And we can basically overlay the data on top of the on top of the solution manifold, and you can see that they basically follow these dark areas of low error, and it happens for some of the subjects. Now note that this is not a model of the behavior. This is just a simple simulation that suggests that if subjects start with a given initiation phase and a given movement time, it facilitates the restoring of the ball rotation at the end. So with that, I'm just going to wrap it up with summary. So we showed that subjects basically modulate the ball phase.
You're going to get kicked out of the room in 2 minutes. Why don't you why don't you go ahead and answer one of the questions at least? So the first question is, could you address the difference between your experiment design and how the wrists and fingers in reality are used to manipulate a wine glass?
Sorry, how the fingers are used to. Can you repeat the second part of the question?
Yeah, so it basically asks, like, could you talk about the differences between your experiment design and how the wrists and fingers are used in reality?
Oh, well, I mean, it would be a different experiment if you have a different grip of the object, definitely. So the experiment design wasn't necessarily to replicate a swirling of the wine, but we just wanted to basically have a hand. I mean, we had limitations in capturing the cup with the algorithm, the shape detection algorithm. So we had to design the cup in a certain way. But I would say that if you were to grip the cup in a different way, then the results probably would be different, just because it's a sort of different configuration.
Okay, I think we'll have to close now because the next session is supposed to start, but I've screenshotted the questions and I'll send them to you. Maybe you can reach out to the attendees later. Thanks, everyone. Thanks for the great session.
Thank you very much.
Thanks to all the speakers.